Hi, and in today's video, we're going to go over active transport. And this is going to be a more complex video. I feel like more high level because Jerry was thinking a little bit higher of himself. As you can see, he put advanced level biochemistry and cell biology. So thermodynamic and cellular equilibrium, so you're gonna get a lot out of this video, and I hope I can teach it good enough. Okay, so there's something called a thermodynamic barrier, and when we talk about active transport, it is an endergonic process, and that means it requires energy coupling to overcome the entropy driven flow of diffusion, and the total energy required depends on the electrochemical potential gradient there is. So the change in G, which is the changing gradient, is equal to R times T times the ln of C2 divided by C1 plus Z times F times the change in V. And the chemical potential we have is the energy required to move a solute from a lower concentration, C1, to a higher concentration, C2. And electrical potential is the energy associated with moving a charged particle, which is the Z, across a membrane potential difference, which is the change in V. And that is the thermodynamic barrier. And then we have the taxonomy of active transport. And there's different types of active transport. And the primary active transport, it couples the exogonal hydrolysis of ATP to the uphill movement of solutes. And then we have a representation of secondary active transport, where it utilizes the stored potential energy of existing ion gradients to drive the transport of another solute. And this happens during the ETC, or the electron transport chain, during cellular respiration. As you can see here, here's the protein channel, and here's the ions, and here's the cell membrane, and the vectorial catalysis are the proteins that act as pump that translate chemical energy directly to physical movement, which is these. I do like this representation, but okay. So primary transport, or the P type of ATP passes, and P-type ATP passes are a large family of cation pumps that undergo reversible phosphorylation at a conserved aspartate residue during the catalytic cycle. So we have 4 different ideas we need to know of domain structure, which contains 3 cytoplasmic domains, A, N, and P, and a transmembrane domain of T. The N domain binds ATP, while the P domain contains the phosphorylation site. Next, we have selectivity. Highly selective for cations such as Na+, K+, Ca2+, and H+. And then we have organella function, which maintains ionic homeostasis. For example, the Ca2+ AT passes the SERCA pumps calcium into the sacroplasmic reticulum. And then we have regulation by ligands, phosphorylation, and inhibitory proteins such as phospholipid, lambin, in cardiac tissue. Hopefully you have a background idea, or this is an introductory idea to when you actually go into the class, because I'm really just trying to give you an idea or what to think about before you go into class, before you actually learn it from a teacher, so you have a existing knowledge and hopefully you can remember it easier. Okay, next we're gonna go into the E1 and E2 catalytic cycle. So as you can see here, we have a figure demonstrating the sodium-potassium pump Na plus to C plus pump. You can pause the video and look at this now. But we have 4 different stages of E1 where the sodium ion binds, and then we have phosphorylation, then the Na plus releases, and then the K plus binds. So the pump right here toggles between E1, which is the inward facing one, and the E2, which is the outward facing one. And phosphorylation by ATP induces the E1 to E2 transition, while dephosphorylation triggers E2 back to E1. Next, we have energetics of the Na plus and potassium pumps. So the pump is electrogenic, which means it exports free positive charges for every 2 imported, contributing directly to the negative interior of a cell. So we have the free Na pluses that goes out, but it takes in 2 potassium ions. And this requires ATP consumption. And in a resting human, this pump consumes approximately 25% of all cellular ATP. And in active neurons, this figure rises up to 70% to reset gradients after action potentials. You can pause the video here to look at this, but I'm gonna move on. Next, we have ABC transporters, which is ATP binding cassette. Okay, so this is one of the largest protein families, and this transports diverse substrates, including ions, lipids, peptides, and xenobiotic drugs. The nucleobinding domains are conserved domains that bind and hydrolyze ATP to drive conformation shifts. The transmembrane domains are variable domains that determine substrate specificity and form the translocation of pathways. And then we have the power stroke, which are ATP binding induces the nucleobinding domains dimerization, forcing the TMDs to open to the opposite side of the membrane. As you can see here, we have a representation of it with coupling helices. As you can see, you see the MBDs binding, and then we have the membrane, the TMDs here. You can pause the video to look at this representation, but we're going to move on to secondary active transport dynamics, which is called co-transport, and it doesn't utilize ATP directly, but it actually uses the electrochemical gradient of the driving ion, which are usually Na pluses or hydrogen ions, to pull the passenger cell upstairs. So we have 3 different co-transport types, and we have symport, antiport, and coupling ratio. The first one is substances move in the same direction across the membrane, like Na pluses with glucose co-transporters. Second one, we have substances that move in opposite directions across the membrane, like Na plus or Ca2 plus exchanges. Or we have something called a coupling ratio, where fixed stoichiometry determines the maximum accumulation ratio, which sees 2 Na plus for every 1 glucose molecule. So you can see the SGLT1. We're going to go into more of that with the case study. So in the small intestine, glucose must be absorbed against the concentration gradient from the lumen into the intestinal epithelial cells. So you can see here the Na plus in the glucose with the SGLT2s instead of 1. And this is called thermodynamic coupling. And the inward movement of 2 Na plus ions down the steep electrochemical gradient provides enough free energy to concentrate glucose up to 10,000-fold inside the cell. So we have the lumen of proximal tubule, the brush border of epithelial cells, and the blood vessels and you see the process which the glucose goes through here, goes through that, and the glucose moves down into the glucose T2, and then that's where the glucose reabsorption happens. But then the Na also moves upwards and goes in here to the blood vessels. But also, don't forget for every free, something has to go out with the exchange. So the Na plus goes in and the potassium goes out. And this system it relies entirely on the Na plus and the K plus AT pace on the basolateral membrane to continuously clear intracellular Na pluses. And to go more into that exchange, we have the antiport Na plus Ca2 plus exchange, where this is critical for cardiac muscle relaxation, and it rapidly removes the intracellular Ca2 plus following heart contractions, like when your heart beats. And the stoichiometry to it is that for every 3 Na ions enter, 1 calcium 2 plus ion is extruded, and this is reversible, as the transport direction can be reversed if the internal Na plus rises or membrane potential depolarizes significantly. And then we have pharmacological applications, and the dioxin inhibits the Na plus K plus pump, which actually raises the internal Na plus, which slows down the NCK and increases intracellular Ca2 plus for stronger heart contractions. Okay, moving on, we have the bulk transport of endo and exocytosis, and this is where large molecules are able to move in and out of cells via vesicle formations and fusions, and this transport requires a lot of energy for cytoskeletal remodeling and membrane bending. So phagocytosis, when the cell eats the large particles of pathogens, where they are fully engulfed by cell extension into phagosomes. And then we have pinocytosis, and that's where the cell, or some people might say the cell is drinking the extracellular fluids and dissolve solutes via microvesicles. And then we have something called receptor mediated. Sorry, I'm a little sleepy today. But highly selective uptake of specific extracellular ligands triggered by receptor binding. And exocytosis is when secretion of intracellular cargo like hormones, neurotransmitters, through vesicle fusion with the plasma membrane. So that's just different types of transports. Oh, we got a lot of. Okay, we have mediated endocytosis. So this is where it concentrates specifically cargo using specialized membrane domains called clathrin-coated pits. And that's where the LDL uptake happens, where a cell collects low-density lipoproteins to extract essential cholesterol, and endosomal uncoupling, where the acidic interior of early endosomes causes cargo to dissociate from its receptor. And then we have finally receptor recycling, where freed receptors are routed back to the plasma membrane via specialized vesicles. So we have a demonstration right here of clathrin-dependent endocytosis, where we have the receptors here. This is like the cell membrane thing around the cell, and we see the formation of the clathrin-coated vesicles here. We have also calcium binding to the AP2 here. If you want to pause it, you can look more in depth to it. But we're going to move on to vesicle fusion, or the snare hypothesis. And this is where this is a vesicle fusion, and it's dried by SNARE, which is a soluble NSF attachment protein receptor with protein complexes, and it overcomes electrostatic repulsion between membranes. The V snares are located on the vesicle membrane. The T snares are located on the target membrane, and the zippering mechanism is where the helical bundle formation pulls the lipid bilayers together, forcing physical membrane fusion, as you can see here. So we're gonna go more into the real world and talk about active transport in human diseases. So these are 4 common diseases. We have cystic fibrosis, and that is caused when there's a loss of function of mutations in CFTRs and ABC ion channel transport, and this disrupts chloride transport, and it results in thick obstructive mucus in the airways. So you have trouble breathing, and yeah. And then we have heart failure with altered exchanges activities with those different ions to abnormal calcium clearance, causing impaired myocardial contractions and cardiac arrhythmias. Sorry about my pronunciations. I'm gonna skip multi-drug resistance because this video is going on way too long. But we have acid reflux, which is a gastric A+ or potassium ion ATP pump hyperacidifies the stomach, and the protein pump inhibitors blocks this P-type ATP paths. So that's something wrong in your stomach. And then we have a summary of everything we went over. So energetics are just coupling exergonic chemical reactions to endogonic transport processes. Mechanics, which are reversible structural state shifts, and then integration, and then physiological impact. And cellular life is a state of controlled thermodynamic non-equilibrium. And I hope you learned something. If you didn't, I hope you just got a basic idea of what active transport and how more things can go in depth. Oh, this is my longest video. I gotta make sure. Well, here's the image sources that I used. I don't know why. I can't. Maybe if I do. Yeah. Yeah, Jerry really messed up my slides. It's fine. I'll put it in the comments where I got my sources from. And I hope you got a good hope you enjoyed this video. If not, shoot me a text message or whatever. Okay, and I'll see you in another video. Bye.